2 o'clock p.m. in Jakarta time, and of course 3 p.m. Brunei and also Malaysia time. So let me just start. Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon, everyone. So let me introduce first myself. I'm Halis Alda from Swiss German University and also from Indonesian Society for Bioinformatics and Biodiversity. So Prof. Han asked me to be the moderator for today's talk for Dr. Lu. Welcome, Dr. Lu. Thank you very much for your time.
Thank you for hosting.
Yeah. So before we start, I would like to introduce you to the audience, of course. So let me just read your brief CV here. So you originally as a biochemist, right, doctor?
Yes.
A biochemist, and then Dr. Lu has ventured into the fields of microbial bioinformatics and later animal and human genomics or bioinformatics nearly 2 decades ago. In addition to his background in molecular aspects of pathogen-host interactions, Dr. Lu's expertise in bioinformatics focuses mainly on the analysis of big genomic data, such as whole genome mapping and assembly for disease variant discovery. So he was a research fellow in bioinformatics and a permanent biocomputing support at the Roslin Institute, University of Edinburgh. By the way, my daughter just accepted at Edinburgh, doctor.
Oh, congratulations.
It's in the neuroscience. Okay, so you were in Edinburgh for about 10 years, yeah?
Yes.
Before moving to University of Brunei, Darussalam in mid 2015.
Yes.
So you are currently a senior assistant professor in genomics and bioinformatics at both the Institute of Health Sciences and the recently formed Institute of Applied Data Analytics,
Yes.
UBD. So my former dean is also at UBD. If you are familiar, know Dr. Muhammad from University of Malaya, formerly from University of Malaya.
Dr. Muhammad from
University of Malaya originally, but he was at UBD, I don't know, for quite a while, but I'm not sure if he's still there or not.
What is his full name?
Muhammad Ghulam Rasul. Muhammad Rasul?
No.
Muhammad Abdul Majid, sorry.
Yeah.
Maybe he's not there. Yeah, he's quite senior. So you are now and head of bioinformatics at the UBD Biomedical Genomics Core Laboratory. So his research now is focusing on medical genomics, particularly that related to population and disease like cancer, rare genetic disease, and pharmacogenomics genomics. Yeah. So today's talk will be on, which is actually, I'm looking forward to listen to your lecture, doctor, because this is definitely not my area, but I'm keen to learn to know more about, well, in this very specific topic, Whole-Exome sequencing reveals the genetic heterogeneity of osteopetrosis in a consanguineous Iraqi family. So without further ado, let's welcome Dr. Lu for his talk today.
Right. Okay. Thank you very much for the kind introductions, and thank you everyone for joining us online today. So let me just quickly share my screen so that everyone can see the slides. Okay, just give me a second. Right. I hope everyone can see the screen all right. Yeah?
Yes, doctor.
Okay, right.
Yes,
okay. Okay. So today I'm going to share with you the works that we did recently in collaborations with our colleagues, with our Iraqi colleagues. And it was really a bit of a coincidence that we came across this project. So one of the Iraqi professors joined another university, UTB in Brunei recently, and one of his colleagues in Sweden was here in UBD as well. And during the conversations, then it came out, and so he was involved in the whole exome sequencing of these Iraqi families, but he came here, so the data was with him, but he didn't really have anyone to help him to look into it. So we then volunteer and says, why don't we help you and analyze the data? And so this is what we are going to share with you today then. Okay. Before we talk about the disease osteopetrosis itself, I thought maybe I'll give you a very brief background on bone metabolisms, because osteopetrosis it is ultimately is a disease of the bone. Okay, so in bone metabolisms, which sometimes is also known as bone remodeling, it's actually a process that is happening all the time, constantly in any healthy individuals, whereby it's basically divided into two processes: the bone resorptions, which remove the matures old bone tissues, and the ossifications, which is the formations of a new bone or depositions of the bone minerals. Okay, the whole bone metabolism process is really important because it's one way that our human bodies regulate calcium's homeostasis. It also helps to repair all the micro traumas or all the micro damages to the bones, so that our bone will remain healthy. And it helps to reshape the bone growth as well. So that's just a very, very quick few words on bone metabolisms. The main types of cells involved in bone metabolism, as you can see here, are the osteoclast, the osteocyte, and the osteoblast here. Okay, the osteoclast is basically the cells that are involved in breaking down the old tissues. The osteoblast is the one that secretes the new bone materials, and the osteocytes basically derive from the osteoblast, the matures, which basically are the mature bone cells that are being trapped in the matrix. Okay, the process itself, the process of remodeling, bone remodeling itself, can be divided into different steps. So what you can see in this figures here. So initially, you will start with a so-called activation steps, where a pre-osteoclast will arrive at the bone at the bone areas, the tissue areas, and then what happens is this will fuse together to form a mature osteoclast at the site where the remodeling or the bone resorptions is going to happen. Okay, and what happens during resorptions is basically the osteoclast will sit on the site, and then it will start secreting acidic materials to degrade away the bone materials, and form a so-called resorption pits. And then as it move on, as then this mature osteoclast would die, undergo apoptosis, and calcium will be released in helping to maintain the calcium homeostasis. Okay, then the next step is the so-called reversal steps, where the mesenchymal stem cells will now come to these regions. Okay, will arrive in the in this resorption pits here. Okay, newly formed pits here. So and they will start to proliferate and differentiate into what we call the osteoblast cells, and these cells will then mature by releasing osteoids, and they form a new non-mineralized matrix. And then later on, finally, it will then sorry it then at the mineralization stage, the calcium and phosphorus will mineralize together to harden the bones itself, and then it will sit there quietly for times until a new cycle will start to kick in again. Okay, so I think that that is a very, very simplified overview of bone remodeling. Right. So, what about the disease itself? Osteopetrosis. I think not a lot of us have come across the disease. Certainly, I wasn't aware of the disease until I was talking to my colleagues. So, I mean, I think a lot of us heard of osteoporosis. Uh, so, this is osteopetrosis is a big like the opposite of osteoporosis. Okay, so it is the disease itself sometimes is also known as a stone or marble bone disease. It is a very rare genetic disease. So it's the prevalence is around 1 in every 20,000 to 250,000, depending on the type of inheritance, the type of osteopetrosis it is. The rarest, the one of the rare one is the autosomal recessive, which is have a prevalence of 1 in 250,000. Okay, so the disease itself is characterized by increased bone density. So what happens is that uh, instead of the in the normal cells you have the osteoclast resorptions, but what happens really in osteoblast in osteopetrosis is there's a failures in the whole resorptions process. So instead of old bone getting degraded away and new bone materials being deposited on it, what happens then in this disease is all the new bone material just deposited on top of the old one, and they started to increase in the thickness and the density of the bones. Okay, so there's basically no breakdowns of the old bones materials there. A number of gene has been implicated in the disease. This include the chloride channels and some of the ATPases, and also a number of transcription factors as well. But the exact molecular mechanisms actually remains very much unknown. So we know of individual genes, the role of individual genes, but still a lot of it are unknowns. Um, and maybe just to quickly talk about the clinical symptoms. So the severe form of osteopetrosis, the patients will usually suffer stunt growth, and they will have anomalies the most striking features that I'm going to show you later on some pictures is actually the bone. You will see that the bone form anomaly. They have a very abnormal morphologies. Some of them they have a very thick skull as well, and because of the thickening of the bone, so they started having they will press on some of the cranial nerve and cause paralysis, and this would then further lead to blindness, facial paralysis, deafness, and even hypo calathemias and anemias. There are three main types actually, three main types of osteopetrosis. The first one are the more common one, which is the very mild symptoms that we find in adult, and it's inherited in the autosomal dominant manners. The second one is the most severe, and sometimes can be quite lethal as well. Usually happens in infants and babies and children, and most of them do not survive beyond adulthood. And this is the malignant infantile autosomal recessive osteopetrosis. And then you have somewhere in between the childhood intermediate autosomal, which can be either autosomal dominant or autosomal recessive. What is really rare are the X-linked one. I think there have only been a few cases reported worldwide on this. Okay, I think that's sort of give you a quick overview of what osteopetrosis is. So let's look at the cases that we were looking at, that we were studying. So these families, this Iraqi-Kurdish family, have a history of osteopetrosis in the siblings. Okay, so what you can see are some of the X-ray and CT scan of the patients. Okay, so you can see the malformations, especially I think that what is really striking is the scars that you see, and the thickening of it. So you see that the brain cavity is being compressed very badly. So in this family, so the parents are actually close cousins, and earlier on they have a daughters who have passed away now. When she was alive, she was actually misdiagnosed as having a thalassemia majors. And then with the two remaining children, if they get older, they start to develop this kind of symptoms, and then they were diagnosed in the hospitals to be suffering from osteopetrosis based purely on clinical features, on clinical symptoms such as the thickening of the pelvic and lower limbs bones. Uh, Also, they were suffering from anemias, and they were partially deaf and also having problem with their eyesight as well. So as you can see, the two children are actually not doing very well. And so back then, they then decided in the university hospitals in this University of Sulaymaniyah in Kurdistan in Iraq. So they decided to do a bit of a molecular diagnostics, and this is what happens. Okay, so they did a Sanger sequencing of five of the more commonly genes that are more commonly implicated in osteopetrosis. So they sequenced the parents, the two siblings, and uncles, which is the brother of the fathers, and also an aunt. So we were told that the cousins, the uncle and the aunt, so the parents and the adults, basically the adults are not showing any symptoms of osteoporosis. Okay, so the result of that Sanger sequencing came back, and they found a heterozygous missense mutation in the chloride channel 7 genes, and this genes has been implicated in the autosomal dominant form of osteopetrosis. And there are many mutations that has been reported and associated with the autosomal dominance. However, um, what the result itself is actually not very satisfying, because it doesn't really explain why the difference in severities between the brother and sisters. So I think that later on, the colleagues in Iraq managed to secure a grant, and so then they decided that they will actually do. They want to know more about what is really going on, so they decided to do a whole exome sequencing of the entire family. Now, by these times, they were doing the exome sequencing. So a new son has been added to the family. So when they finally got to do the whole exome sequencing, so the family then consists of the parents, and the son was 18 years old already. The daughters was 13 years old, and then they have the 1.5 years old boys as well. Okay, so they did 100 base pair whole exome sequencing, and this was done by a company in Germany. And then so the result came back, and this is basically from this step onwards, this is where we got involved. Okay, so then what we did is or was we took all the reads, we map it against the human reference genome version 38, with the decoy sequences included, and then we code variants using the GATK gold standards protocols, and then we filters off some of the low quality variants, especially those with a Phred score of less than 20 or read depth of 4 or less. And then we annotate the variants using ANOVA, the program ANOVAs, and that by comparing data informations from different databases such as DB-SNPs from NCBI, Ensembles from EBI, and the EXACT, which is the one of the biggest consortiums that have studied thousands of exomes data across the different populations. So that was the upstreams analysis. Now, what happens after that? After we have got the variant file or the VCF file, So in order to identify the genes that are causing the disease, so we then design a workflows and analysis workflow where, first of all, we ask ourselves the simplest questions. So what are all the genes that has been reported uh, wherever, regardless whether they're in GWAS or in literatures or in some sort of a genetic disease databases or whatever. So we decided to search very thoroughly all the genes that were involved in osteopetrosis. Okay, and then we found a number of genes. So initially, so I think the highest numbers that we have seen from different sources were about 20, well, less than 30 genes, but we managed to add on another 20 plus genes. So at the end, we ended up with 56 genes. Okay. And let me quickly jump to the next slide, and then I'll come back to the remaining step again. Okay. So this is how we did our how we search for the genes that were involved in the osteopetrosis process. So after we got the list of the genes, we to make sure that the genes are actually all involved in some sort of involved in the potentially involved in the disease. So we did protein-protein interactions using the strains network analysis, and as you can see here, the different colors here representing the different process or steps in the in bone remodeling. So most of our genes that we found and are actually involved in the bone remodeling process. So that gave us a lot of confidence about those genes that we have. Okay, but you can also see a number of unconnected outline genes here. So these genes we have double check again. So these have been mentioned in some of the literatures, but perhaps, well, we decided actually just to include them, although the evidence are a bit slim, a bit weak here. So, but we still decided to include them. Okay. So once we have those number of genes, so what happens then? We then further expand the 56 genes by looking at all the genes that are involved in the in regulating those 56 genes, and all the genes that were involved in some sort of interactions with those 56 genes. So at the end, we expand the whole network to more than 300 genes, and we fit these genes into our variants where we started to do the filterings. Okay, then what the first thing we do is then we look at all the non-synonymous variants that are present in these genes. Okay, and once we found them, what we do is we look at we then break it down into indels that disrupt the coding frames, and then we further then look for variants we have which has alleles frequency of less than 0.05. Now, because can I just remind you, this is a rare genetic diseases, so we don't expect the alleles if the person is suffering from osteopetrosis, that the alleles should be present in a high frequency in the populations. Okay, and in addition to that, we also look at the splicing side. Now, the exome sequencings that the German did for us, and if you know anything about whole exome sequencing, it's never exactly the sequencing never really fall exactly within the exone's boundary itself. There are always flanking regions of the exome that were also being sequenced. It's just because the way the probes were designed. So we decided then to include plus minus 7 base pair within the splicing areas of the exons, just to check if there's any variants that will contribute to splicing errors as well. And once we have done that, we have identified the candidate variants, then we do some further analysis, include protein modelings. Okay. And ideally, we should also do Sanger sequencing just to double confirm it, but we have not done that yet. And so, and in addition to looking into the potential genetic variants in osteopetrosis, what we have decided to also do, to make the analysis more comprehensive, is we decided to actually search to see if the three children are actually suffering from any other rare genetic diseases because of some new de novo variants that has not been or that are very rare or has not been reported anywhere else. Because we have the whole exome data, so why not make full use of it? Why not look into it? And so that perhaps with some of these variants that we identify, we can maybe even explain additional clinical phenotypes that we saw in these families. So here are the results then. Okay. Um, so we ended up with a total reads of about 7.7 gigabase pair. That's from the 5 family members. And then after mappings, we calculate the coverage. So that gave us about 38 folds of coverage. Now I believe the Germans. So when they sent the sample for sequencing, they were actually supposed to be sequenced 50x, but we get 30x, which is not bad. It's good enough. But then when you look into the mappings on the exon itself, and that's what we mean by on target. So basically, we exclude all the mapping outside of the exon, so the flanking side. Yeah. So then the coverage come out to be about 26%. But that's not really a big concern again. I think anything above 20%, 20x is probably pretty good already. And we were really happy actually to see that the quality of the sequencing is really good. That we found that about 99.97% of the reads can be mapped onto all the known exons in the human genomes. Okay. And so with this sort of a pre-check or quality check on our mapping, so we're quite happy with that. And then we start looking into to identify all the genetic variants. So what you see are the number of genetic variants that you found in the different family members, and of this, they share about 23,000 genetic variants together. So which means that it's exactly the same, and various overlapping number of variants shared by the different families here. Okay, so then we move into the in-depth analysis. Okay, so this is what we got at the end. Okay, so we found that we identified about 27,000 single nucleotide variants which fall on the target exons. So again, this is the area, just the exons areas. Okay, and of this 27,000, 26,500 are reported in different databases, especially either in exact or in DB-SNPs. And we found that about 500 or 600 of them are actually novel that has not been reported in any others databases. And then after we go through all the filterings that I mentioned earlier on, looking at non-synonymous, the flanking side, and applying the minor allele frequency, and we come up with so first of all, so when we look at the main gene itself, we managed to narrow it down to about 65 variants. Okay, that and some of the variants that you can see here. By share, we means of this, how many of them are shared by the siblings. Okay, and what is really interesting for us, we managed to actually to narrow down from 27,000 variants down to about 4 variants. Okay, and we managed to identify actually all these 4 genes, which are all related in some way to bone remodeling, and hence give a very good confidence about their roles in osteopetrosis. So let's look at them in a little bit more details. Okay, the first SNPs that we're going to look at is the actually, this is nothing new. This is, in fact, the one that was reported earlier on during using Sanger sequencing. So as you can see that this is the Sanger sequencing results published earlier on. And we basically reconfirmed that with our NGS data, with our Whole-Exome data, which we are really happy. So it's basically giving us more confidence that our WES is working fine. So CLCN7 genes is a chloride channels that are involved in the exchange of protons and chloride, and this is one of the most widely implicated genes in osteopetrosis, and it can cause either autosomal recessive type 4 or autosomal dominance. So, but because what you see here among the siblings here, they're all heterozygous. So the results would then support the hypothesis that the children is actually suffering from autosomal dominant osteopetrosis, as the Sanger sequencing has done. And the next variants that we found is in the genes LRRK1. This is a gene which encodes a leucine-rich repeat kinase, and they're involved in GDP, GTP binding proteins, and they play a role, has been known to play a role in regulating the bone mass. So there has been some studies. In mice, that when they have a deletion mutations, the mice actually shows very severe osteosclerosis located at the end of the bones. So then, but however, looking at this, the mapping status here, now we see that the story is not very clear cut. Okay, in this particular SNPs here, we see that only the mothers, the daughters, and the young son, the young baby boys are carriers. They are all heterozygous. Okay, so the elder son and the fathers homozygous reference, which means they're not the carriers. Now we don't really know because they're really the genetic disease informations for this particular gene is missing. So all the information we got is from the mice. So we don't really know how the inheritance or how the mutation of this is exactly going to impact on the disease itself. Okay, but it is potentially genes which may impact future generation of this family. The same goes with the another genes, the ATP6VOB. So this is actually a veculose ATPase. Is one of the domains, the VO domains of the ATPase. Now the other domains of the ATPase is also well known to cause osteopetrosis, and I think this is the first time we actually, as far as we are concerned, I have not seen it in any reported literatures. This is the first time we are reporting here these variants, potential variants that in osteopetrosis patients. Okay, but there has been some GWAS studies that associated with bone mass density, and in zebrafish, the homologue of the genes has been mutants of these homologues has been known to cause different eye diseases as well. Okay, with our families here, Iraqi's family here, what we saw actually the fathers and the daughters are carriers in these genes. Now again, we don't really know if the mutations is actually having any deleterious effect on the daughters or not. Okay, so that will require a bit more investigations. Now, what is really most interesting for us and satisfying for us actually is the discovery of these new mutations in the genes SNX10. This is a genes that has never been reported. Again, it has never been reported anywhere else as well. There have been some one or two cases of osteopetrosis in different part of the world where SNX10 has been reported to be causing the disease. But in these particular locations, these particular mutations, no one has reported it yet. Now, what is really interesting for us is that we actually saw that for the son and the daughters, the eldest son and the daughters, they are homozygous for the mutating alleles, which resulted in the stop codons of these genes SNX10. So this gives us a very very confident diagnosis that these two siblings is actually suffering from homozygous recessive form of osteopetrosis. So a little bit more about the SNX10. Okay, SNX10 encode for proteins of 209 amino acid, and it is a protein that is involved in the sorting and membrane trafficking and endosomal homeostasis. So we then did the protein modeling to try to predict what is the impact of that stop codons. Okay, so this is the models that we did, and so what you can see here is the wild type models of XNX10. It is actually a dimers. It exists in the dimers format, and we know they are the important arginine 16s and they form a hydrophobic contact with proline 157 here. Okay, and then hydrogen bond on glutamate acid 158 on the change B. Okay, on another change of the on the wild type. Now, when you have that mutations that we saw here creating the stop codons, the end result is that basically you're abolishing the contacts between the in the monomer itself and also the potential contacts of the dimers. And because of these disruptions, we then predict that the functions of SNX10 in osteoclasts are going to be severely affected, and there are probably no protein-protein interactions. Okay, so what is the role of this SNX10? What are the genes they are actually interacting with? So we know that from the studies done in 2013, and there's been a model has been built. So they're suggesting the SNX10s by interacting with the VATPS. Now this is another domains. Okay, the study was done using another domain, not the V node domains that we identified just now. So this is the V node domains. So that study was done with the V1 domain. So they were saying that the SNX10s is involved in the trafficking of the proteins between that comes through the VATPS. So so you can see then the number of different genes that are expressed in osteoclasts here then will have an impact on the protein remodeling process. Okay, so we were really quite happy about this particular finding that and I think we are quite confident to tell the clinicians that for that particular two brothers and sisters, their previous diagnosis are probably wrong. That the two brothers and sisters are most likely suffering from the autosomal recessive form of the osteopetrosis. Okay, so those are the genes that we identify. Sorry, the variants that we identify in in osteopetrosis. And as I mentioned earlier on, we also look at we also try to identify any new de novo variants in the three children, and hope and in the hope that if we may be able to identify any genes that may explain some of the phenotypes. Okay, so these are the results that we found. Okay, so the way that we our definition of de novo as variance here, actually I think I should have explained this earlier on. So our definition of a de novo as variance is actually those that we think are violating the Mendelian's rules of inheritance. Okay, so which means then, for example, looking at different genotypes. So you have the dominant or allele genotypes, and so for example, you have heterozygous in fathers and AB, capital A and then small a's in the mothers, which is the recessive, and you have the possibility of four different combinations, which is the classical biology class that everybody learns in school. But when they deviate from that kind of combinations, then this is what we call then Mendelian violations, and we treat those as potentially de novo assembly. Now you're going to ask that could those Mendelian violations actually due to sequencing error? Yes, they could potentially come from sequencing error. Hence, we are very stringent and very careful when we choose them. So we make sure that they have sufficient sequencing depth or coverage. We also make sure that the quality of the reads, the quality of the base, and also the quality the quality of the variant codes are very good, very high. And so this is then the result. So what you see here is the daughters, the son, and the young babies with an unknown status. You stand for unknown status in terms of his whether he's suffering from osteopetrosis or not. Okay, so at the end, after much filterings, we managed to find a number of de novo variants in the three children in the three siblings, which is the numbers actually it's actually not uncommon at all. I mean, in any individual's humans, this is probably the kind of numbers that you will see. But what is worrying is actually this de novo as variants here, they all have a deleterious predicted deleterious effects. Okay, so what are the genes here? Okay, so we have a genes here which is linked to potentially linked to cutaneous T cell lymphoma. So they're involving cancer. Another genes which is ribosomes receptors, and this genes has been known to be promoting has been shown to promote colorectal cancer progressions. Okay, and then we have another two genes, the mucin the mucin 12. This is a family, a member of the mucin families, which has been implicated in epithelial renewal and differentiations. And we have one other genes which may not have any functions. This is a pseudogenes. Okay, so these require more investigations. I think we have to be careful with some the findings here. First of all, they should be Sanger sequence again, and then even better is we then have to look at the clinical data, try to understand if any of these de novo variants could potentially actually explain some any others conditions in these children. Okay, so I'm going to wrap down what the thought here. Okay, so in conclusions, we found a stopgap mutation in X and X10 genes, which I think most likely explain the osteopetrosis in the especially in the elders, in the two elder siblings, and this mutation is an autosomal recessive forms, instead of the autosomal dominance osteopetrosis that was suspected early on from the CLCN7 Sanger sequencing. And we also identified three heterozygous mutations which are potential carriers of the disease, and they could potentially impact on the disease itself as well. So our studies is actually the first time that any study has shown that the disease to be polygenic in nature, which means that you have a number of non-osteopetrosis genes, or a number of genes that are involved in bone remodeling, that harvest some mutations in there. Okay, so I think this opened the doors for us to actually look into futures, more refined molecular resolutions or molecular diagnosis of the disease, so that you can classify the disease more accurately and see if actually when you have say one genes affected, you know, what is the severity and what are the symptoms, and then when you have this multitudes of symptoms, what are the different genes that are actually involved. Okay, so I'm gonna end here, but can I then just have another few minutes actually before I open for questions. Oh, here, by the way, here are my collaborators. So this is my groups of people working on this project. My students, Mirza Asmi is the main persons that has been doing most of the analysis, and then my collaborators in UBDs, and then Professor Bistons, who came from Iraq, and he's our contact with our Iraqis clinicians, and he was the one that did the whole exome sequencing. And then we are funded by UBD and also the Kurdistan Institution for Strategic Studies and Scientific Research. Just before I finish this, I just want to advertise. We are actually recruiting soon these positions. Please do keep your eyes on the website of UBDs at least. So if you're interested, you can contact me and talk to me. We are also recruiting postdoc as well. Again, please get in touch. And if you don't know, and actually to make it a bit more attractive to you, so I thought maybe I'll give a very quick introduction about our universities and our institute. Okay, so as you can see, our Institute for Health Sciences, is located in a very nice environment. We're not far from the sea. We have a very nice sunset here that you can see. Or sun, actually, I'm not quite sure whether this is sunset or sunrise. So we are a very young university of 35 years old, and we are also a rising university. So we will rank 35 among the under 50 years old universities by QS ranking. And this we have also just about to start using our new biomedical genomics core labs. As you can see, the kind of facilities that we have. So we have Illumina Next machines and all the related equipments that going to support the machines. And so the lab is very nice. So we're going to start using this very soon, and we hope more people will come and join us. And also, these are the type of works that my group is involved in, and that give you some ideas what are the kind of the, research that we are doing. So the main purpose is basically we are hoping to build the foundations for precision genomics healthcare in Brunei. So we look into different aspect of it. So we do comparative genomics by comparing genetic variants from Southeast Asia and the rest of the world with our Brunei's genetic variants, and then we look at genomics of the of regulatory functions. We look at pharmacogenomic, cancer genomics, rare diseases, and then we do a comparison between baseline and disease genetic to give us a better understanding of the disease, so that it would lead to precision genomic healthcare. So those in the gray are the areas that we are hoping to start soon. Okay, I think I'm open for question now.
Thank you very much, Dr. Lu, for a very impressive work. And of course, there are so many that have been revealed, and I think there are more to be revealed from the research. So it gives
Yes.
us a very good insight about the disease, although it's quite rare, but I think it will be very important discovery that might relate to any other disease. So I would like to open for Q&A session for some question from the audience that have joined us today. Thank you very much for all the participants today. So please raise your hand if you have any question, and I will give to the three question. Yeah, three question to person would be wonderful, because we don't have much time anyway. Go ahead.
I mean, you can email me later on if you have any other questions as well.
All right. Okay. If there is no one question, then I have one question, Dr. Luke. So while we are waiting for other participant to have question to answer question to ask some question. So I was just wondering about this. Is there any reverse, well, what you call effect on? Well, this is osteopetrosis. Do you have any or data evidence the now related to the osteoporosis? What is there? Is there any reverse fact about this disease? And that's my first question. The second one, is it typical to Kurdistan family or any other maybe tribes or race? Like, for example, cystic fibrosis, for example, is very typical for Caucasian.
Yeah.
But what about this disease? Thank you, doctor.
Yeah, yeah. I'll answer your second question first. Okay. The osteopetrosis seems to have a higher prevalence in the Middle East and Mediterranean areas. So this is a well-known fact. But osteopetrosis has also been reported almost everywhere in the world. In Japan, and because it's quite a rare disease, and it only gained more attention in the recent year. And I've seen more and more reports coming out from China as well, especially over the last few years. And there is also a concentrations. It's well known. I think there's an area in Greece, if I'm not mistaken, where they have quite a high prevalence of that disease. But so all these, a lot of them, I'm not saying all, but a lot of them are the result of consanguineous relationships, so intermarried within the families. Okay, the second question is a very good question. So I've been thinking about that as well, whether we can apply the knowledge we gain from osteopetrosis and help it and maybe in some way reverse the effect of osteoporosis. But unfortunately, I think most of the genes are different. The genes implicated in osteopetrosis and osteoporosis are different. But I can see your arguments there. Say, for example, one potential way that I can see is, as we understand more about the whole osteopetrosis process, the whole bone remodeling process, can we instead then look for example, instead of knocking in and out of a gene itself, what about looking at the regulatory elements, regulatory regions of the genes in bone remodeling, in bone resorptions, okay? And look at where to maybe either up or down regulate the expression of these genes, and maybe then it will improve the osteoporosis, the bone decay process. I think that is something that I would imagine if we have enough data, enough genetic status, or even combine the two the disease together, do some sort of a large scale genetics GWAS studies, it could potentially come up with something interesting.
Okay.
Yeah.
Thank you, doctor. Thank you very much for your explanation. Any other question audience, ladies and gentlemen, maybe? Because I have another question. Okay, Miss Akinola. Okay, go ahead. Let me just unmute you. All right, go ahead, Miss
Okay.
Akinola. Or okay,
All right.
Mr. Akinola. Yeah, okay.
Sorry, it's a mister. Thank you, Dr. Zenlo.
Hi.
That was a beautiful presentation. My question is, I just want to confirm or to find out, did you guys carry out any functional studies on those genes that was found, the four genes, especially the XN, X10, which is
Not yet,
loader?
not yet. We are actually going to apply for a grant to do functional studies. Most likely, we would like to perhaps do some sort of an in vitro cells assay first.
Okay, in such a way that maybe you can knock out, maybe in zebrafish or in mouse, that you can knock out
Yeah,
the gene and
yeah,
see
yes,
and see if you can actually get the same um, effect
yes,
or the clinical symptoms what we have in those
yes,
family in the animal. Okay, so
yeah.
that would be a good.
That will be the next step to do. Yes.
Okay, all right. Okay, thank you.
Thank you.
Thank you, Dr. Akinola. Any other question? We still have some few minutes, precious minutes. No. Do you see any chance, doctor, uh, well, we are heading to the precision medicine for every single genomic disease, but do you see at current situation any other drugs or medicine that can possibly overcome this disease, or
The,
maybe gene therapy, probably?
yes, there have been some trials actually on gene therapies
Okay.
targeting the CLCN7 genes, but I think the success has been limited so far. But I would imagine then that's probably the way to go. But again, I think the disease can be difficult, and I can see why it has not been very successful. If we look at our data here, it's suggesting that the disease itself is really quite heterogeneous in nature. So if you just
Right.
simply targeting one gene or targeting one single variant, you are unlikely to really make a big improvement in the disease.
Right. And that's the reason why I think it will be quite difficult, because it's vary among
Yes,
individuals, right?
yes, yes.
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 Okay. Thank you. All right. Any other question, ladies and gentlemen? Because this is a very interesting topic, actually. Even I have another question, but it's impressed me when you try to extract from the 27,000
Yeah,
and then end up with only 4
yeah.
variants. That's something very, very actually intriguing to me. That's I would like to learn about that more, actually,
We, we
later
are
on.
we, we are we are open to collaborations. We are quite
Right.
willing to collaborate with anyone.
Right.
I think it is actually very important. The more data we have, the more powerful our analysis will become, and the more confidence
Yeah.
our hypothesis will be.
Yeah. Well, my background is infectious disease, microbial
Okay.
pathogen, so it's
Yeah.
more like, yeah.
Yeah,
Okay.
we also
Yes.
have an interest in infectious disease here. So
Okay. Yeah. Right.
so we would like to start into that as well. So one of the things that we are thinking of at the moment is actually, I mean, everybody is doing COVID-19, so we have to do
Right.
COVID-19 as well. So yeah,
Okay. Do you have BSL-3 facilities? I saw your very nice.
not in our universities, but
Okay,
in the
but
Ministry of Health they have. Yeah,
all right, the ministerial
yeah,
have it. Okay, all right,
yeah,
very interesting.
yeah.
So because my training was in infectious disease. So okay, we still have 1 minute left. I still open one question to everyone who would like to some question, because this is literally we are all heading to precision medicine, right, doctor? I
Yes,
think this is one. It's just the open the windows to how we can reveal this genetic disease from the very early on, from the symptoms, because what I see the symptoms is quite vary, like blindness, deafness, so many symptoms that
yeah.
maybe one way or another relate to any other disease, for example. So that's very interesting too. Okay.
I think this
Yes.
also demonstrates to us that in the past we have always been trying our best to simplify diseases, but that's probably taking the wrong approach. I think as we get
Right.
more data on genomics, we start to realize a lot of diseases is actually contributed by multiple genes rather than a single genes.
Right, because as my background as well, doctor, I work with protein interaction
Yeah,
when I was doing my PhD. So I think at some point, maybe nearly all proteins they interact one another at
yes,
certain point in
yes,
time.
probably yes.
Yes, I
Yeah,
think that, yeah, because even for infectious disease, for
yeah,
example, in either flagella or biofilm assembly,
yeah,
the proteins are there, but they interact one another,
yeah,
and when should be downregulated, when should be
yeah,
upregulated. That's
yeah,
really depend on different circumstances.
yeah.
Okay, if there is no more question, I would like to, as on behalf of the Perdana University SDX, I would like to thank Dr. Zenlu for your very nice discovery and work, and showing a lot of collaboration with different institutions. And of course, I would look forward to your new establishment with so many. This I think UBD is heading to a very good direction. And for participant, everyone, thank you very much for coming. And also, really, it's my very good opportunity to um, know you, Dr. Lu, and also for the Perdana University. So I
Thank,
would like to hand over to this to the host, and thank you from me. Good afternoon.
yeah, thank you for moderating and thank you for hosting.
Thank you. Yeah.